you, David. Our next speaker is Richard Finn from UCLA's Geffen School of Medicine, where he's associate professor, and also from the Jonsson Cancer Center at UCLA, where he is co-director of the Signal Transduction Program. And Rich will talk about some of his experience in clinical development and early signals of efficacy. Rich?
Thank you very much, Mace. Thank you all very much for the invitation. What a distinguished group of speakers. I feel a little out of place. But we'll talk about this maybe from assessing early clinical signs from even the preclinical step. And when I was a fellow, this was a breaking story. This was a very exciting story. Patients with advanced lung cancer got a pill, and within days, their tumors were disappearing. And the literature was filled with diagrams like this. You know, you have the EGFR kinase, and simply we could block that kinase activity with a pill, and then all these downstream things will fall apart and will cure lung cancer. But needless to say, a lot of the development of EGFR inhibitors really were limited to models that were designed to show what they wanted to see. That is to say, they were clearly very EGFR addicted, one of the most popular ones being the A431 cell line. It wasn't even a lung cancer cell line, but an epidermoid line that has very high levels of EGFR and was very exquisitely sensitive to EGFR inhibitors. But clearly the EGFR pathway is much more complex than those diagrams. There's a lot of networking, interactions, and the key, obviously, to successful development of this class of drugs would be to be able to identify patients where you simply knock off the head and everything else will fall into place. And that is clearly not most lung cancers by any means. And needless to say, and I think you've heard it earlier today, here's one of the randomized studies of gefitinib. Compared to placebo at 2 different doses, there was no benefit. And there was then phase 3 studies with this compound that were negative. And even though there were patients who clearly got a benefit, the drug was only approved a few years ago in the United States at least. And given there's another part of the story with erlotinib. But needless to say, they did not know who to give it to until retrospectively. And you're very familiar with these papers that came up after the failed phase 3 study. So it raises the question of how do we do better? Could we have predicted this failure? And moving ahead with new agents and new targets, how do we approach that? How do we know what's going to be a success and what isn't going to lead to success? And here just showing in 2010, 10 years after early studies, they finally had a prospective study that showed benefit in patients selected for those mutations. So the approach that we've taken at UCLA, and I should comment, I've been at UCLA for a little while. My mother had breast cancer. I started working with Dennis Slayman in the early 90s. Obviously a very exciting time in cancer medicine. HER2 had just been discovered, and I was very attracted to the program because while it was a scientific program, it was also very clinically focused. And as I was finishing my clinical training, it was very easy to come back to Dennis's lab, and at the time, dealing with this EGFR failure, how could we do it differently? And also going back, because it has been some years, I guess, is that we were identifying that malignancies, that certainly in solid tumors, did not represent one disease entity. We knew that clinically, and using now breast cancer as the prototype, one of the first diseases to undergo whole genome expression arrays to identify what we called hormone receptor positive, HER2 amplified, and triple negative really represented many molecular subgroups of the disease. And similarly now in lung cancer, it's no longer non-small cell lung cancer, but mutated disease. And building a preclinical platform that might inform the development of compounds in the future. And this preclinical laboratory that we set up now exists of over 600 cell lines, which have all been molecularly characterized and integrating new technology for mutations, gene expression, copy number variation, and then working with industry to get molecules to look at in this laboratory in an organized way across histologies to try to correlate response biological effects with molecular characterization, and ideally to inform clinical development. To do this type of approach, when a drug's already in phase 3, we're not going to be able to make an impact. And as we go back to our non-small cell lung cancer panel and look with gefitinib or erlotinib, you'll see that out of 60 cell lines, there's 3 or 4 that are exquisitely sensitive, and of course they have EGFR alterations. And wouldn't that have been nice to know before failed phase 3 studies? So over the past several years, Dennis and I have been going around to various industry partners to get molecules to look at in this preclinical platform. And so one of the molecules I'll be talking about, the one I will be talking about, is one I've been very involved with, and that's palbociclib, or the CDK4/6 inhibitor, now known as Ibrance. And this study, this paper was in Science a few years ago, and it's an interesting paper that highlights some of the challenges in drug development, certainly in big pharma, because this is a molecule that was actually synthesized in the 90s, and then kind of got lost, I think, in the shuffle a little bit in acquisitions and big pharma. But ultimately, also knowing where to go with this compound. So palbociclib is a first-in-class inhibitor of CDK4 and 6. You'll remember that cyclin-dependent kinases are a large family of proteins that regulate the cell cycle. The Nobel Prize was awarded in 2001 to Hartwell, Hunt, and Nurse for their pivotal work in defining the importance of these proteins. And specifically, there had been earlier CDK inhibitors. Most of them were pan-CDK, flavopiridol being one of them that was very cytotoxic, preclinically and also in the clinic, and never went very far. And it probably didn't go very far because of the compound and its PK properties, but also not identifying a group of patients who would most likely benefit. And clearly, the idea here is that you block CDK4/6 kinase that interacts with cyclin D. This blocks hyperphosphorylation of the retinoblastoma gene product, and therefore it would induce a G0, G1 arrest. But obviously, the cell cycle is pivotal to every cancer. So how would we best identify patients to move ahead into the clinic? And again, just commenting that we knew breast cancer was a very molecular heterogeneous disease. And if you only took 1 or 2 cell lines and said this represents breast cancer, you could very easily miss important clinically relevant observations. But still, despite all this great biology, in the clinic it's still approached as 3 diseases. ER positive, which represents the biggest group, which clearly has benefited by the introduction of anti-estrogens, the most important management thing in that disease. Dennis's seminal work in HER2 and trastuzumab, and for the 25% of patients who have HER2 amplification. And then if you don't have those 3 markers, you're defined as the absence of those markers, and that's a very bad group of patients because only chemotherapy is available. So this has now become, I think, an important observation for drug development besides just CDK4/6 inhibitors. And what you see here from work done with Dennis and others at UCLA in collaboration with Pfizer, we looked at this molecule not making any assumptions. We just looked in a large panel of breast cancer cell lines, looking at their sensitivity. Here they're color-coded by their subtype, whether the luminal ER-positive group, HER2 amplified, or those that we consider non-luminal or would represent the triple negatives, and the most sensitive cell lines are here on the y-axis down low. And you can see clearly an enrichment for those that were ER-positive versus those that were non-luminal or ER-negative. So right away, we've generated a hypothesis to move ahead into the clinic. The other approach would just be take advanced breast cancer, give the patients the compound, and then try to sort back, where do we go with the compound? We went on to show preclinically, given the importance of anti-estrogens in ER-positive breast cancer, show that across several different cell lines, across large dose response curves, that the compound interacted in a synergistic manner. We also showed that we block RB phosphorylation and induce a G0, G1 arrest. So this led to a phase 1, phase 2 study. Initially, this was a proof of concept study. That's what this study was geared to show, that our laboratory observation could hold up in the clinic. It was very important, I think, from some work I've done in other diseases, to have a randomized study, not to overread single-arm phase 2 studies. And it was initially 150 patients, open label, palbociclib and letrozole versus letrozole alone. Palbociclib's phase 1 dose of 125 milligrams, 3 weeks on and 1 week off. Came from a prior phase 1 study. Keith Flaherty led that study. But as this study was accruing, the idea, and actually Mace has said this several times, that to have a negative study, you can really kill a compound. It's hard to resurrect. And while the most important biomarker that came out of the preclinical work was just having ER positivity, the study was redesigned and incorporated prospective assessments for cyclin D1 amplification and P16 loss. Alterations we know that occur in breast cancer, but weren't necessarily, from the preclinical data, most important for selecting patients who benefit. So to show how this evolved quickly, we initially presented data at an impact meeting in 2012. Did not really make a big splash at this time because a lot of people were skeptical. And here in a small number of events, and this was just in that first part that were ER positive, HER2 negative, we saw PFS went from 6 months to 18 months. Pretty remarkable, and a very quick early separation. And to me, this was validation of our preclinical observation. This was not an accident, because we had a good understanding of where we're going with this compound and why it is working in this group of patients. We also went back and looked at some of the biomarkers, quote-unquote biomarkers, but we were looking at cyclin D1 and P16 in the second part of the study that was ongoing accrual, and we showed here that whether you had cyclin D1 or P16 alterations or not, there was still a dramatic benefit of the compound. So at that time, this study was stopped. We did not need these additional biomarkers, and the decision was made to launch a phase 3 confirmatory study. And I say confirmatory because, remarkably, I would say, and many of the people probably involved in the decision are in the room, but, palbociclib got an accelerated approval last year on February 3rd based on this study. And this study, at the end of the day, had 165 patients. So that means about 85 patients got the new therapy, palbociclib. Yet it got an indication for the first-line treatment of ER-positive breast cancer in combination with letrozole, the first CDK4/6 inhibitor to be approved in cancer medicine. And this benefit actually stood up in both cohorts, whether you were just ER-positive or ER-positive with the cyclin D1 or P16 alterations. And as I mentioned, it got a breakthrough therapy designation. And I think much to the FDA's and the community's approval was that the phase 3 study looked exactly the same. We got a 10-month improvement, just over 10-month improvement in the phase 2 study of 10 months to over 20 months, and in the phase 3 study, which was just published a few days ago, again, over a 10-month improvement in PFS with the addition of palbociclib to letrozole. And this has moved fairly quickly, I think. Our collaboration with Pfizer started in 2007. We initially shared that data a few months later, moved to a phase 1 of letrozole and palbociclib just to show there's no drug-drug interactions and it was safe. In 2009, the phase 2 was open, and we presented that data in 2014. It got a breakthrough therapy designation, was approved in 2015 in the United States. And then global approval is occurring as we speak. And I should comment that there's other CDK inhibitors in development. This compound from ribociclib from Novartis went from phase 1 to phase 3 in the same exact indication and has nearly the same exact results. Hazard ratios 0.58 versus 0.56. And again, has ongoing studies with fulvestrant where palbociclib has been approved as well. And to be complete, abemaciclib, a compound from Eli Lilly, again, in the same exact indications from phase 1 to phase 3, with anastrozole or letrozole, as well as with fulvestrant. So now we have 3 CDK inhibitors all going after the same indications, and in reality likely will all meet their endpoints, some of them have, in that indication. And just to comment, initially, abemaciclib looked in all comers of breast cancer, and reassuringly, they're seeing activity really only in the hormone receptor positive group. And this compound has a breakthrough therapy designation as well, earlier, just about a year ago. And, you know, everybody was skeptical, right? And we can go back to this paper, and I think it relates a little bit to Wolfgang's last talk about PARP inhibitors. This molecule, you know, we did not submit our randomized phase 2 study to the New England Journal of Medicine because we didn't think they would take it because of this experience. You know, here was a compound that was looked at, it was touted as a PARP inhibitor, looked at in with combination with chemotherapy in triple-negative breast cancer. It looked very promising in phase 2 with a survival advantage, yet failed in phase 3. And I would argue the reason for that is, one, they didn't know what this compound was doing, much different from the CDK4/6 inhibitors, where there was a very strong preclinical package identifying a group of patients where it benefited, its synergy with antiestrogens, as well as some biomarkers for response. And I just like to show this slide. This comes from the TCGA, where they profiled breast cancers and highlighted, you know, pathways of interest. This came out in 2012, well after our initial work with palbociclib, but identified alterations in the RB pathway and how there might be some sensitivity markers in luminal disease or ER-positive disease, cyclin D1 alterations, as well as alterations in CDK4, high expression of RB. However, if we had just limited our patient selection to these markers, yes, we would have had a positive study. But we clearly would have missed the other 80% of patients that might benefit from a compound like this. So we always need to be very humble with all our molecular data and how that actually impacts clinical development. So in conclusion, I think you've heard, and I would say that the greatest improvements in cancer patient care has come from the translation of laboratory observations into practice, into clinical trial design. And preclinical models can help us design those studies to generate hypotheses-driven phase 1-2 studies rather than just hypotheses generating. And I think certainly with CDK inhibitors, we were lucky in that ER positivity contains the biomarkers that are needed to get a positive study. But as these drugs move into other histologies, I think we'll have to be a little more careful in how we select patients. I just want to acknowledge all the clinical investigators that helped push across PALOMA-1, the phase 2 study, as well as the patients, their families. You know, Dennis has gotten a lot of support from Revlon and the Department of Defense, which helped support the preclinical platform that we use to generate this data set. And acknowledge my colleagues at UCLA TRIO and also highlighting our great collaboration with Pfizer. Thank you for your attention.